Hello, everyone, and welcome to today's ASBMB webinar from Signature to Systems Multi-omics Architecture of MASLD. I'm Chenmin Jiang, ASBMB's Assistant Director of Publications, and I'm very pleased to be moderating the session today. This webinar is part of ASBMB's Breakthrough webinar series, which is sponsored by the ASBMB journals. Specifically, today's session is brought to you by the Journal of Lipid Research, one of our esteemed ASBMB journals, which has been at the forefront of lipidomics and metabolism research for over 60 years. Before we dive in, a quick reminder, you're welcome to submit questions at any time using the Q&A feature at the bottom of your screen. We'll address them during the dedicated Q&A session. After the presentation, this webinar is being recorded, and the recording will be available on the ASBMB website. It's now my pleasure to introduce today's speaker, Dr. Silvia Sookoian. Dr. Silvia Sookoian is a senior research scientist at the CONICET, head of clinical and molecular hepatology, and dean of health sciences at the Maimonides University, Argentina, a graduate of the University of Buenos Aires. She's a physician scientist dedicated to translating scientific discovery into clinical care. Her research focuses on the genetic, epigenetic, and the molecular mechanism driving the pathogenesis and the progression of metabolic dysfunction-associated steatotic liver disease, MASLD. Dr. Sookoian's work bridges the gap between laboratory innovation and patient-centered outcomes in the field of hepatology. Without further ado, please join me in welcoming Silvia. The floor is yours.
Thank you, Chenmin. Hello, everyone. I'm very happy and pleased to be here today. So we'll start to join you with this fantastic initiative of the Society. I'm very grateful for the Society, also the Journal of Lipid Research for this nice invitation. So I have no any financial relationship. I must say I'm an editorial board for different journals, and also I'm associate editor of Journal of Lipid Research. So today I would like to share with you some innovative approaches in MASLD. I'm asking, are you seeing okay the screen? Is everything good, Chenmin?
Please share the slides now.
Yeah, I'm doing. 1 minute, 1 minute, 1 minute, 1 minute, 1 minute, 1 minute, one more, one more, one more. Okay, I think it's good. It's good. Yeah. Good. Okay, so I just telling you that I would try to summarize some innovative approaches to understand the biology behind MASLD. So to break the ice, I would like to refresh quick concepts. The different layers of omics in any eukaryotic cells are, of course, genomics. And when you involve the chromosomes, the chromatin, the nucleosomes, you go further with epigenomics. And also, we have different layers with transcriptomics, proteomics, metabolomics, and other omics. I will go further in this conversation. But the thing is, when you do multi-omics, the concept is you integrate all levels of biological data with clinical data. And that's the beauty of doing this for understanding the human diseases. But there is something more, because when you have all this information together, you need to go ahead with very specific analysis of networks at different levels. So the situation gets complex and complex. But still, this is the very interesting part. So you see here, this is really the key of understanding why this is important, because at the very basic levels of any cells, there are many connections and correlation between genes that have very similar interaction profiles. For instance, from the mitochondria for functions such as RNA processing or protein folding, everything is connected. So this happens, you can imagine, in a very simplistic experimental model, such as the fungus, but you have this in humans as well. So what about MASLD? First, I would like to refresh the concept. MASLD has been recently redefined and renamed because there are many, interpretations from the patient side and also from the physician side, and then we need to do something with the disease to be more inclusive with all the different phenotyping of steatotic liver disease. And MASLD is one of the component of steatotic liver disease. You can read this in Journal of Lipid Research. We wrote with Mario Rinella about the new name. But the thing is, we have three elements to understand the biology behind the disease. Number one is steatosis, and hepatocytes accumulate fat inside the cell. Number two is when the fat gets more and more, you have a second situation, that is the inflammation. This is very simplistic, but we are progressing with this concept. But the thing is, when your fat is accumulated in excess in the cell, this causes stress and injury to the hepatocytes. And then when this progresses even more, you have scar tissue that is fibrosis. But these diseases, MASLD, has other two very important features. One feature is phenotypic complexity, that the phenotype of the disease can progress to MASH, that is steatosis, and then cirrhosis. And also it has inter-individual variability, meaning that not all patients will progress with the same speed, or not every patient will get MASH or cirrhosis or hepatocellular carcinoma. And also the impact at the epidemiological level is very important, because almost 30% of the population, adult population, will have MASH very soon. And also this is the number one reason for transplantation, at least in the United States. So the concept is a very prevalent disease, it's a very huge health problem worldwide. But coming back again to the concept of omics, what is really true that in the field of MASLD specifically, there were two specific omics that were very much developed across the years, that are metabolomics and lipidomics. And in this cartoon, you can see, in this plot, you can see all the genes that are classified according to novelty and according to importance. There are most of them, probably 90% of them, are all targets involved in lipid metabolism. And this was work done many years ago, and now recently another genetic study focused on MASLD again confirmed that most of the locus involved in the disease are specifically enriching molecular functions, including lipid binding and sterol transfer activity, and cellular components of enrichment of these locus are in terms of lipoprotein particles and lipid protein complex. So let's go to dissect in a way each omic layer, and start with number one is genomics. MASLD has the same, like other complex diseases, the same heritability that is around 25–30%. But we need to go back to the past and remind us the first genome-wide association study on MASLD in that time, NAFLD, that this study was focused only in non-synonymous variant, very clever strategy to start. And this shows us a hit. This dot is the variant in PNPL3 that was specifically very much high in Hispanic population and was only associated with hepatic fat content. And then we replicated this finding in a small but yet very well characterized sample of patients in Argentina, showing that the case of the variant was associated with the disease severity, not only with the fatty liver, but also with the phenotypic NASH. Now it's called MASH. So in this table, you will see the main drivers, the most studied genetic variants. Again, all of them, except the glucokinase receptor, are involved in lipid metabolism. And the effect is very also diverse as well, but number one in terms of higher effect is PNPL3, that has a 3-time risk of having more advanced disease. And on the other side, you have the protective variant, that is a spliced donor variant that has very, compared with the other, strong protection against the disease severity. But also we have other variants, for example, in the case of TM6SF2, that has a very dual and opposite role in the disease, because while confers one allele, confers risk against, this is an all publication. That's why you see the all nomenclature in the cartoon. While one allele gets protection against the disease, the other alleles cause cardiovascular disease. And this dual opposite role is very important, because while this variant has an important role in the susceptibility of the disease, is not a good target because this dual opposite role. Yeah. So here again, we see the key targets, and this is to remind us that these targets, while are located in lipid droplets, most of them that are also in very interesting components of cells, such as endoplasmic reticulum or Golgi compartment, but again, all of them are involved in lipid metabolism. And this is why this is very important, because this is the way we translate the genomic knowledge and the omics knowledge to the drug discovery, because today we have two different approaches, one using PNPL3 and another the HSD17B13 protein, that they are specifically designed with oligonucleotides against the disease. So the concept is you can use either small interfering RNAs or oligonucleotides, and there are many different combined with GalNAc to allow these compounds enter to the liver. And now we have in phase 1 or 2 different clinical trials using this strategy. This is a very clear and very important demonstration that genetics can be translated into drug discovery. For example, in the case of PNPL3, the concept of doing this in the clinical trial, the concept is the information came from the very preclinical studies showing the importance of the protein and the messenger RNA, and then the investigator moves to the phase 1, and they found a very favorable safety profile, which is good to go ahead. And also they found that the target protein was very well engaged, showing a decrease in the messenger RNA and no signs of complication, but the inflammation in the liver was reduced using kind of the targets that mimics the inflammation, such as IL-3 or C-reactive protein. And this is a very, very clear demonstration of using personalized medicine because the clinical trial was only indicated for patients carrying the risk allele. And this is a case of how they show the reduction of PNPL3 protein from the baseline using immunohistochemistry. And the another strategy for moving genetics to drug discovery is focus on the variant, the protected variant in HSD. And in this case, we know that the A insertion allele is associated with decreased risk of chronic liver damage and also MASH with approximately 40% reduction in the odds of developing disease progression. So the idea is you have here immunohistochemistry of patients, real patients having 2 copies of the A allele. And the idea using this as a molecular target is to mimic the protective effect of the variant. So the idea is knock down the messenger RNA expression. So that's the background for designing a clinical trial, again, using, and in this particular case, ARO. The idea is a small interference therapeutic DNA. The trial is now in the very, very early stages, but again, it's a very important proof of concept how the genomics moves into the field of drug development and is translated for the care of patients. So you can find the results of a trial in Journal of Hepatology. Is a very small clinical trial, but still is promising. So we reach 2026 with a very specific portfolio of drugs that are all interventions based on dominant or proximate mechanisms of action. So all are molecular targets, including the oligonucleotide drugs. You can find this in a recent review. So each target has a molecular justification of being specifically there. And also the latest oligonucleotide drugs are also based on what information we have in disease biology, but using specific approaches. These are, again, as I said before, oligonucleotides or mimic RNA, but all of them, of course, on a very early stage, such as murine or in that case, primate models. But again, we need time, but this is the new generation of drugs. So there are other fields that are very interesting in the latest years, that is the using single-cell transcriptomic. I will go with this very quickly. It's just to remind us what exactly means doing single cells. When you do bulk analysis, you mix all the cells together and you have an average bulk of what is going on in the tissue, for instance. But when you go ahead with single cell, what you are doing is to specifically select the population you are looking for or are trying to study and understand what's going on in the transcriptome in that specific cell population. And that's happening in the liver. It's very interesting. Early models show us that there are very specific cell type proteins and transcriptional programs for each of the cells in the liver, not only for in the case of hepatocytes, but also in liver endothelial cells or hepatic stellate cell. Any subtype has their own specific transcriptional programs. Hepatocytes, for instance, involved in energy metabolism, xenobiotic, whatever you name it, and the same for the other kind of cells. There are many resources to understand this in detail. We recently collect actually we worked during the pandemic doing this reference atlas, but it's very comprehensive. You have many details inside. But an example of an interesting intersection between single cell and genetics is that the main targets I mentioned before really didn't have very specific difference according to cell types, and many are expressed in cells of the immune system, and also there are many differences in these specific targets according to the different tissues. But what's really interesting is that these specific targets have different transcription according to age and according to sex. This is also very important when we talk about drug designs. And another resource we have talking about omics is the instruments that allow us to understand the drugability of any target we can choose. For example, in this case, STAT3 in blue, you see the very high drugability. Drugability means the likelihood that a biological target can be modulated by any small molecule of biological drug. In contrast, we have in blue in this chart glucokinase receptor, a very low drugability. So this is important also to have in mind for drug discovery. What about proteomics? And this is an example of using system biology and proteomics to understand the disease biology. In this particular case, we collected all the proteomic studies involved in MASLD and MASH, particularly involving the circulating proteome, and we saw after doing enrichment and overexpression analysis that most of the proteins that are downregulated and non-regulated in cluster associated with cholesterol efflux, high-density lipoprotein particle clearance, and reverse cholesterol transport, and those who are upregulated in the circulation were those in clusters involved in leukocyte cell addition, inflammatory response, and complement activation. And what is interesting is that when we focus, for instance, in analysis of protein domains, there were some protein domains that probably we wouldn't see if we were doing this proteomic individually. For instance, some group of chemiokines or some group of C-type lectin domains, or for instance, this SUGI domain that is very particular and very fancy as well. So the interesting thing is this is doing an ad for that system biology give us as the specific omics analysis. And we know that MASLD is not alone in the body, and most of this characteristic of the latest study is doing the analysis of a connection between MASLD and other diseases as well. This is, for example, a nice FIWAS on MASLD. So MASLD is inserted in a very specific cluster of non-communicable diseases. So for this, having this in mind, we try to understand by doing a large proteomic study to understand whether MASLD severity predisposed patients to systemic organ damage and extrahepatic complications. So for doing this, we started with a target approach using a very small sample of proteins, and then we escalated this, replicated in the UK Biobank, and we select the specific overexpresses or downregulated proteins in the blood by combining different omics technologies from liver expression, bulk transcriptomic, single cell sequences databases, or spatial proteogenomics, and then contrasting the hypothesis in organelling and modeling. The idea is that by doing all of this, we reach a protein called ADGRG1 protein that is involved in organ damage, but also originates in the liver because this was expressed only in patients at risk of MASH. But also when we look at the very prospective data from the UK Biobank, we discovered that this protein originated in the liver was a predictor of mortality, severe liver complications, type 2 diabetes, lung disease, and ischemic heart disease. So it was a predictor of extrahepatic complication. But what is interesting as well is when we analyze this protein, particularly in the protein atlas, we noticed that it was associated with autoimmune disease, cancer, cardiovascular infection, besides MASLD. So this is a very interesting example how you can use the analysis of the dynamics of the protein, for instance, to predict treatment response. And this is the case of using this protein, as I mentioned before, in the case of the protein signatures associated with type 2 diabetes. So again, ADGRG1 is the one of the 30 top proteins linked to type 2 diabetes, but what is even more interesting is when you analyze the proteins that are modified by any, in this case, therapeutic strategy against type 2 diabetes, this protein is again, among the most significantly modified proteins under therapy. So this is a very, very interesting concept. So you can monitor the effectiveness of a therapy by selecting a protein that is able to change with the treatment. Another layer of omics is the metagenomics. So in 2020, we started to question whether the liver tissue of our patient can have, in this particular case, DNA from bacteria. So we have done this study, was published in Gut, and we find that the liver tissue has a community of diverse bacterial DNA, that is not despreciable, is quite high compared with other tissues. This was replicated in further studies as well, but we pioneered this question. We confirmed the technique using different approach, not only next generation sequencing, but also PCR. And also we uncovered that the liver tissue, have a high concentration of LPS, particularly patients with MASH. So we found a group of bacteria that were associated with severe histological features. One of the group, DNA derived proteobacteria, were elevated in the tissue, and other group were downregulated. But I'm telling you this because I'm trying to connect the next slide, that is the importance of gut bacteria in the epigenetic modifications, either DNA modification or RNA modifications. The idea is, the concept is that the leaky gut and the DNA or any product of the bacteria are strong modifiers of fatty acid oxidation, insulin resistance, and of course, in the case of MASLD, are involved in the progression of the disease. So with this background in mind, we again came back to our collection of tissue in our biobank, and we connect this liver microbial DNA composition I mentioned before with the liver acetylome and with the 5-HMC methylation global levels. This is a kind of tricky experiment because you need to use fresh nuclear extracts from the liver. That's why you need, to collect the liver in very specific conditions. But again, we measure the total activity of deacetylases and acetyltransferases. And what we found was a very significant correlation between some kind of bacteria that were the bad bacteria and the good bacteria with the level either HATs or HDACs in the circulation. And the concept is, when we went to the profile, the metabolite analysis, we uncovered, by doing another statistical analysis that is not naming, such as causal mediation analysis, we uncovered an indirect effect of bacterial DNA on the degree of steatosis. That is, this indirect effect was mediated to the level of HAT in the circulation. And the decrease in the enzymatic activity of HAT in the circulation with and in the liver is associated with an increased production of bacterial-derived butanoate. So there is a confirmation that the presence of DNA, the bacterial DNA in the liver tissue is not inert, is very important to trigger some of the feature of the disease. And also, we try to connect the liver microbiome with the genome. And in this case, we try to understand the connection between the host genetics of the major drivers and the tissue microbiota. And we found that by doing a polygenic risk score, that almost 7% of the genus level taxa variation was explained by this polygenic risk score with the major variants I mentioned before. So another important resource we have when we are talking about omics is the combining data-driven biomedical with omics. The concept is you need to integrate data on health and disease with longitudinal data, meaning it's much better if you collect different times in the lifetime of the disease biology and integrate this. When you integrate this with any resource, either machine, unsupervised or supervised machine or deep learning, this is a powerful instrument to understand disease risk prediction of any disease, of course, disease subtyping, biomarker discovery, and understand molecular insights. So this is very important because at the end of the day, it can also inform you about treatment response. Why I tell you this? Because coming back to MASLD is like any other complex disease, very heterogeneous. And why is very heterogeneous? Because MASLD is overlapped with other diseases of a metabolic syndrome, type 2 diabetes, arterial hypertension, obesity, dyslipidemia. So you have very diverse subphenotypes, yes? Combination of MASLD with obesity, combination of MASLD with hypertension, you name it. But also you have many environmental factors that modify the disease along with the genetics. But also there is population variation that is completely unrelated with disease physiology, but again influence the disease physiology. You have different causative biological processes, microbiota, genetics, etc. So the disease is heritable and there is also epigenetic heterogeneity. So this is the concept of understanding the molecular heterogeneity. You see only a very small part of the disease when you study the disease. So there are many resources to disentangle this complexity. One of these is, for example, you can use any deep learning machine or strategy. In this particular case is cluster modeling to declassify the different subcomponents. When we collect data back in the days of different publications and try to understand the subphenotypes. And we realize that in each combination, you have different dominant and different patterns, molecular patterns that are very common to the specific combination. For example, in the case of arterial hypertension and MASLD, we have a very interesting cluster associated with defects of DNA repair and also BGF. So this is also more recently a very nice study doing using this strategy of polygenic risk scores in the UK Biobank. The investigator disentangled two different subtypes of MASLD with different prognostic implications, one confined to the liver resulting in more aggressive disease and the other with higher cardiovascular risk. Another approach in Chinese population, they use whole genome sequencing, proteomics, phosphoproteomics, lipidomics, and metabolomics, and they uncover three different subtypes. One, when associated with negative feedback on steatosis, the other focus on the infiltration of macrophages in liver cirrhosis and another involved in HCC carcinogenesis. There are things that we are missing here, that is the research is a bit delayed, that is the use of pharmacogenomics. For example, in this particular case, one of the two drugs approved for the MASH treatment is resmetirom. Said 70% of the patients are non-responders. There are many factors, but genetics can be an explanation if you, for example, target understanding variants in the transport or variants in the drug target specific. So to end this conversation with you, my view is that the omics evolution in MASLD passed through two big waves. The first wave gave us genomics, transcriptomics, and epigenomics, and in a way was a very illuminated way to understand the genetic predisposition, to realize that the MASLD is very heterogeneous and there are different levels of biological organization. And then we have the second wave, very interesting way, because by doing the multi-omics integration, we had metabolomics, metagenomics, and proteomics into the understanding, along with very robust computational, statistical, a different instrument for understanding this data integration that came us to the point of having novel drugs. So as an example of how we integrate, in our recent review about genomics of MASLD, we are saying that for making a good precision stratification of the patients, we need to join different layers of data. And this is exemplified for understanding the genetics. You not only focus on the disease, but also in other syndromics, on rare diseases associated with MASLD. So thank you very much. I hope you enjoy this. I'm very, very happy to take questions with you.
Okay. Thank you, Silvia, for the insightful presentation. So we now have around 5 minutes for questions. So
Okay.
I'd like to remind everyone that you can submit a question using the Q&A feature if you haven't done so yet. So our first question is, can we manage the MASLD through yogic intervention?
I don't understand. I'm reading the question, but I don't understand what you're trying to ask. What you mean exactly?
Maybe the person who can elaborate more, can
Yeah,
you include your another question
yes.
there, just to explain what you really mean for the yogic intervention. And at the same time, I have one question. So, what advice would you give researchers entering the multi-omic field? What's your advice for the new incomers into this field?
Yeah, this is a very, very nice question, because the first things you have to do actually is to understand the very basic of omics. You need to, elaborate your research question very carefully and choose your omics layer very carefully. But if you manage to integrate data, you probably will get awesome results. So yeah, the advice is to answer the specific question, to use the specific instrument or model for your question. I'm coming back to the question of, I reading you mean yoga?
Yeah.
Yoga. Okay. Yeah, I must confess, I'm not an expert, but any strategy for our patient is good. Of course, yoga, yeah, of course. Dietary intervention, yes. Exercise, yes. There's no restriction about this, but I'm not an expert.
Okay. So
The
thank
first
you.
wave, I mean the third wave, yeah, I'm seeing Chengmin. Okay, this is special omics, yeah. The special omics is in the second wave. I mean, for the reason of time, I didn't show you, but yeah, special tell us about how, I mean, to choose very specific cells in the right moment in the right place. And there are many latest discovery telling us that in the liver the transcriptional programs are zonated. So each zone has their own transcriptional program, and this is very important. This is very important when you choose your research question. Yeah, the spatial omics, I think it's not in the third one, it's in the two, but this is very interesting as well. Yes.
Thank you, Sylvia. We have a last question.
Yes.
There are studies of longevity mouse model that show resistance to MASLD. How can this model be incorporated in drug discovery?
Well, I mean, I must say that there is some in the latest years, there much controversy about using rodent models because depending of the experimental model you are using, they do not replicate the human disease. But there are some strategies that are very good for very specific questions. When you use a knockout model, for instance, this is a powerful strategy to understand the reason of any protein or any target involved. When we are talking about fibrosis, the things are getting a bit complicated because the model indeed, even manipulating the model with dietary approaches, the model do not replicate the disease biology, and particularly talking about spatial omics. But anyway, it's a good start. My concept is when you use an experimental model in mice or rat, you need to try to replicate this in humans as well.
Okay. Thank you, Sylvia, and thank you everyone for submitting the thoughtful questions. And Sylvia, I'd like to hand it back to you and for any final thoughts or key takeaways before we conclude today's session.
Okay, thank you, Chenmin. I mean, first words are for the audience. Thank you so much for having me here, and also for the society, and also for people that I cannot see, but I can imagine there are many young people behind the screen trying to start in the MASLD field, but this is very exciting field, and also it's very nice that you can do something connected with omics and send your nice paper to Journal of Lipid Research, because we will happy to see your research on human studies as well. So thank you again for having me here. Hope to see your new studies in the future. Thank you so much.
Thank you so much, Sylvia, and for your time and expertise today, and to all of you joining us. Be sure to keep an eye out for next month's webinar in ASBMB's Breakthrough Webinar Series, and have a great day. Thank you.
Thank you. Bye-bye.